Okay, this is chapter 32, just blood. So there's obviously more components to chapter 32, but I'm just going to do a recording of just this last section that explains components of blood and function of blood and a few other things. So in terms, it talks about blood typing as well. So in terms of the function of blood, right, transports gases, nutrients, waste products, antibodies, hormones throughout the body, right? Basically, it's a fluid connective tissue, right? And its function really is in transport. Helps combat pathogenic microorganisms because you have white blood cells within there, right, that fight disease, that are part of the immune system. Helps maintain water balance and pH. So in terms of, you need to make sure that there's the appropriate concentration of water and also salts and also ions in order to maintain that proper pH of the blood. Regulates body temperature, also carries platelets and factors that ensure clotting to prevent blood loss. Okay, so you should know those functions. So in terms of functions of the blood, there they are. And so now if you look at the composition of the blood, you can see that it's made up of both plasma and formed elements, right? So plasma is a sort of fluid matrix to which all of these formed elements sit within. And what this is showing you here is if you take a tube of blood, just like if you go and get blood drawn and they put them into those tubes, right? Those glass sort of test tube looking things, then they spin them down in the centrifuge and basically separate it out this way. So what you are left with is just plasma here at the top, which is about 55% of the blood is plasma, and then about 45% is the formed element. So this is the red blood cells here, and this little tiny little thing here are the white blood cells. If you talk about the makeup of plasma itself, mostly water. 90–92% of plasma is water. Okay, so this helps maintain the blood volume and transports molecules. There are also many plasma proteins, so they'll help to maintain osmotic pressure and pH. We talk about osmotic pressure, talking about ion concentration and also the sort of concentration of water versus ion concentration. Some examples of some plasma proteins: globulins, fibrinogen. Fibrinogen is particularly important in blood clotting. These globulins in fighting infection. Salts, about less than 1%. And then also gases, oxygen, carbon dioxide, obviously are carried around in the blood. Nutrients are carried around in the blood as well, and as well are wastes and also hormones. In terms of formed elements, red blood cells, and we saw this when we looked at our slide of blood in lab, right? So red blood cells, essentially, they are there to carry oxygen and carbon dioxide around, and they do this by, having the protein hemoglobin on them, and that's where the actual oxygen is attached to. White blood cells, there are many different types of white blood cells. You guys don't need to be able to identify different types of white blood cells, but remember these are immune cells, okay? And each one of these has a particular unique function. Platelets are involved in clotting. Okay, so again, if you talk a little bit more about those red blood cells, like I told you, their job is to carry oxygen and carbon dioxide around. They don't have a nucleus, okay? And like I said, they contain hemoglobin. Hemoglobin is made up of 4 protein chains, and each is associated with an iron-containing group. Okay, that's why it's super important that your iron levels are appropriate, right? So if you have a decrease in the amount of red blood cells or the amount of hemoglobin caused by a decrease in iron or what have you, it can lead, it's called anemia. Okay. So super important that your iron levels are where they should be, because it's necessary for your oxygen to be carried around in the blood. Okay, these red blood cells are made continuously in the bone marrow, right? And we talked about that red bone marrow when we mentioned a bone in chapter 31. Okay, so we talked about the spongy bone, right? But also bone in the skull, the ribs, vertebrae. Red bone marrow is there as well. And also, there's a hormone called erythropoietin, which will stimulate red blood cell production. So no, this is one of the hormones that was sort of used in doping. I believe Lance Armstrong, this was the hormone that he took, but there's also other ways of increasing this hormone. So there's other types of drugs that you can take that will increase levels of erythropoietin, right, to then lead to more red blood cells. If you have more red blood cells, you increase the oxygen carrying capacity of your blood, and therefore you'll be better as a endurance athlete if you can carry more oxygen around. Okay, The other part of this is talking about blood typing, okay? And we talked about blood typing when we, back in, or you should have, if you in Fundamentals 1 or General Bio 1, when you do the genetics chapter, usually you'll talk a little bit about blood typing. So blood type is determined by the presence or absence of a surface antigen. Okay, so this means on your red blood cells, this antigen is there. Antigen is just a protein that sits on the surface of the red blood cells. Okay, and there's two kind of identifiers. So there's the ABO system and also RH system, right? So if you have antigen A on your red blood cells, then you're type A, okay? So we're going to talk about this. And then what happens is whatever antigen you have, so if you have the A antigen on your red blood cells, then your body is going to have B antibodies, okay? And so B antibodies in your plasma can cause something called agglutination, and essentially that's a cross reaction when you have these antigens and antibodies that will meet, and you can end up with organ damage, right? So again, if you have B antibodies circulating in your blood and somebody gives you type B blood by mistake, like for a blood transfusion, it will result in this process called agglutination, and you basically get this coagulating of the red blood cells, and it leads to organ damage and basically ultimately to death. All right, so if you look here again, if you're blood type A, that means you have the A antigen on your red blood cells. And then what antibody you're carrying around in your plasma is the B antibody. If you're blood type B, it means you have B antigen on your red blood cells. And in the plasma, you have antibodies against A. If you're blood type AB, that means you have both the A antigen and the B antigen on your red blood cells, and therefore you have no antibodies in the plasma, right? If you had antibodies to A and B in your plasma or one or the other, then your own body would be attacking its red blood cells, okay? If you're blood type O, that means you do not have any antigens on your red blood cells, therefore your body has antibodies to A and B circulating in the plasma. Okay, so you should know this, okay? I could ask you on the exam, if you're blood type B, what type of antigen do you have on your red blood cells? Okay, so this is basically illustrating what I just said, okay? So if we look here, so this is showing you a matched blood transfusion, right? So here's your type A blood of the donor, right? And you can see these A antigens, okay? And then anti-B antibody of the type A recipient. So this means that the recipient getting this type A blood also has type A blood, which is good, right? That's what you hope. And so therefore, since they have type A blood, they have antibodies to be circulating around. Therefore, when you give them a type A blood with these A antigens on the surface, this antibody cannot bind, okay? And then therefore, there'll be no reaction and everything is all well and good. Everything's fine, okay? However, if you give this person the wrong type of blood, okay, there's going to be a problem, right? So this is showing your type A blood donor, right? Type A has these A antigens on the surface of the red blood cells. In the recipient, they're actually type B, so they have A antibodies in their blood. What happens when you give that type B person type A blood, you can see that this antibody, right, this B antibody, this A antibody will bind to the A antigens, therefore producing this reaction called agglutination. You can see this clumping, and basically leads to blood clots and organ failure and eventually death. Okay. The other thing is we talked about the ABO system. There's also RH system as well, and so you either can be RH positive or RH negative. This talks about the fact where this is kind of a, I guess, a sort of unique, kind of rare thing that might occur. I'm not going to ask you about this, but just to kind of go over it, just because you might find it interesting, I'm not going to ask you about this on the exam. But it says if the mother is RH negative, right, and the father is RH positive, it's possible that the unborn child might be RH positive, right, but the mother is RH negative. If RH positive blood cells leak across the placenta, right, that would be super dangerous to the mother because she's Rh negative. So the mother will produce these anti-Rh antibodies, and therefore it would attack that, right? So anti-Rh antibodies cross the placenta and attack the red blood cells of another Rh positive baby in a subsequent pregnancy. So just because you're going to end up having these antibodies around. And so you can prevent this by giving an Rh negative woman basically a immunoglobulin or antibody injection, okay, within 72 hours of bearing this Rh positive child. Anyway, don't worry about this. Like I said, this is just sort of extra information here of something kind of rare that can happen, but as long as you know about it, you can prevent the problem, right? If you don't know about it, it can be really, really dangerous. Okay, so again, the Rh, really, I'm only going to ask you about the type A, type B blood, type AB blood, and type O blood, what that means, okay? Again, don't worry about the Rh system, but you either then are Rh positive or Rh negative. Okay, so for white blood cells, so that was all pertaining to the red blood cells, right? So if we talk about white blood cells, again, these are immune cells, okay? Most of them are larger than the red blood cells, and I'm sure you saw this when you looked at the blood smear under the microscope. They contain a nucleus, which stains really darkly purple, and you can see that on the slide. They lack hemoglobin. Again, they're important in immune function, right? So it says inflammatory response, and there are 5 main types. You do not need to know the types of white blood cells, okay? So don't even worry about this. I'm not going to ask you about granular or agranular leukocytes, okay? By the way, leukocytes is another name for white blood cells. You do not need to be able to identify neutrophils versus eosinophils or basophils, okay? That's for another time. Just know that their white blood cells are immune cells, okay? Same thing. You don't have to worry about this. You don't have to worry about the different types of white blood cells, okay? The last component that we'll talk about are platelets, okay? So platelets are involved in clotting. They result from fragmentation of these megakaryocytes, which are a type of sort of blood, I guess you can think of sort of precursor cell in the red bone marrow. And so again, they're fragments of cells, right? So they're not really cellular. There's about 150,000 to 300,000 per cubic millimeter of blood. They're involved in clotting, like I said. So a blood clot will consist of platelets, also red blood cells, and then also fibrin threads, okay? And so there's a particular enzyme called thrombin that will activate, that will essentially convert, well, that sort of activate fibrin, or I guess, right? They'll convert fibrinogen, which is another protein, into fibrin. And then those fibrin threads will wind around the platelet plug to basically provide a framework for the clot. So you can kind of think of, when we talked about connective tissue and they have these fibers in the matrix, that's kind of what these are going to do. They're sort of thread-like proteins that will provide the sort of framework for this clot to occur, right? And then there's another protein called plasmid that will destroy that network. You don't need to know the details of this process, okay? But it involves platelets, but there's also other proteins involved in the clotting process and hormones that essentially, that will kind of, or enzymes that will sort of activate this process. Sorry, not hormones. Okay, so this is kind of showing you, if you look at blood clotting, what I just mentioned. So here, blood vessels punctured, right? And you're going to have red blood cells coming out. Platelets will congregate in that area where that, puncture was to form this plug. And then basically it'll initiate the sort of formation of that fibrin, right, in these fibrin threads. And that'll trap all the rest of those red blood cells there so that it basically heals or seals up that puncture. Okay, so that's what this is showing you here. Again, you guys don't need to know the details of blood clotting, all right? That's sort of an extra thing, okay? So I'm not going to ask you about the steps in blood clotting. We've been talking about this, we talked about this throughout the kind of throughout the cardiovascular system lecture, okay? So this really isn't new. Some of the details are a little bit new, but anyway, this has to do with a little more details about capillary exchange, okay? So we know that in the capillaries, this is where you're going to have exchange of nutrients and of gases, right, of oxygen and carbon dioxide. The walls of these capillaries are very thin, right, because, we talked about this, they're simple squamous epithelium because you have to get exchange to be able to occur. If they were super thick, then nutrients and gases wouldn't be able to cross. Okay. Now, I want you to just look at this picture here again. Here's the same thing. I don't expect you guys to know these details, but I think it's important to kind of just be aware of how this happens, okay? So again, it's really diffusion, right? So it's the solutes diffuse into and out of the capillary according to their concentration gradient. So it's not any, sort of active transport or anything like that. Oxygen and nutrients will diffuse out of the capillaries, again, based on a concentration gradient. Carbon dioxide and waste will diffuse into the capillary based on concentration gradients, right? So meaning, well, I'll show you a picture in a second. All right, and then it says substances leaving the capillary to contribute to interstitial fluid, right? So remember I mentioned, we mentioned lymph and we mentioned lymphatic vessels, and that these, the lymphatic system basically picks up this excess fluid, which you can call interstitial fluid, and then it returns it back into the blood, okay? And so this is what this is showing you here, right? So one, it's showing you a couple of things, right? This is the arterial, right? The arterial, okay, small artery, this has oxygenated blood, right? And then now this is going through, the capillary here. This is capillary, and then eventually to a venule that will then return this unoxygenated blood back to the heart, right? And so basically this is just kind of showing you that at this end, at the arterial end, when you have higher blood pressure, okay, you're going to have oxygen and, again, amino acids, water. These are all leaving, again, based on concentration gradient here, right? So at this end, you're going to have oxygen being released. And then obviously now here, you're going to have carbon dioxide coming in. So this was kind of me. I talked about the concentration gradients. Some of the movement of these molecules also has to do with sort of the pressure set up inside here as well. But bottom line is the movement of things like oxygen and carbon dioxide and glucose has to do with concentration gradient, okay? And so they're going to move from high concentration to low, right? So here, there's lots of oxygen in the blood, not a lot in the tissues because it's delivering it there, right? Or in the interstitial fluid. So oxygen flows out. Here, now you don't have a lot of oxygen in the blood. The carbon dioxide will then move into the blood so that you can then take it back to the lungs to be exhaled. And this is just showing you here, these capillary beds. Again, we saw this all throughout the cardiovascular lecture. It's also showing you, though, here you can kind of see these lymphatic capillaries that are embedded in here as well. And again, these are going to pick up that excess fluid, that excess interstitial fluid, and then bring it back to the blood vessels. Okay, and that's it.